Hello, everybody. Here is Dr. Amr Zahir, and this will be my third lecture on general embryology. Um, People have always been concerned in knowing how they originate, develop, and were born, and why some people develop abnormal. Ancient views of human embryology. The ancient Egyptian believed that the soul entered the child at birth through placenta. That was their thought long time ago. And this was the thought about the function of placenta as the entrance gate for the soul to the fetus. Ancient India's idea have been recorded long time ago concerning the development of the embryo. It states that from conjunction of blood and semen, the embryo come into existence. During the period favorable for conception after a sexual intercourse, they call it Kalada. Kalada, they call it as a one-day-old embryo. After remaining several nights, it becomes a vesicle. After the fourth night, it becomes a spherical mass. After a month, it becomes a firm mass. After 2 months, the head is formed, and after 3 months, the limb region start to appear. This is a little bit close what happened in reality, but it's not exactly accurate. In the Middle Ages, embryology in the Middle Ages, Leonardo da Vinci made accurate drawing of the pregnant uterus containing a fetus. It's very accurate. And they thought at that time that the baby starts from a sperm. The whole baby starts from a sperm of a man. And this is an illustration of a sperm containing a tiny baby that eventually will grow up. But it appears to be wrong, of course. This was not our beginning. Here is our beginning. Something completely different than the shape of adult baby, adult human or a baby. Completely different. In fact, embryos have a tail and gills slit in early stage of development. But why it have to be like this? Why the human embryo look like this with gills and tail? Because at early stage of development, human embryo or a specified human heart here will resemble the heart of a fish, not necessarily Nemo, any fish, with only two chambers, an atrium and a ventricle. Okay, and would grow up to be atrium and ventricle like a fish. Fish have additional conus and sinus venusus. And here is circulation in a fish with two chambers heart, one ventricle, one atrium, with conus arteriosus and sinus venosus. Again, here is the heart of a fish, and here how it gets oxygen from the water through the gills. So the embryonic stage of a fish is very simple, from single tube to two-chambered heart with a conus going to the gills and sinus venosus. Frogs are a step forward in evolution. They are amphibians, and separation starts here in the atrium, only in the atrium, not the ventricle. So the frog will have three-chambered heart, right atrium, left atrium, and one ventricle. And here the embryological stage of heart of the frog, from a simple tube to three chambers heart. Turtles, a step forward, the interatrial septum that separates the right atrium from the right atrium will grow a little bit to the ventricle, but without complete septation of the ventricle. So we'll have a one ventricle with incomplete septum and two atrium. And here it starts from a single tube to the three-chambered heart of a turtle. But they have two systemic arches, right and left. Crocodiles, a step forward in evolution. Here, the septation in the ventricle is complete. So we have a four-chambered heart, right atrium, left atrium, right ventricle, left ventricle. But still have dual aortic arch system. Mammals and birds. Here is a complete septation in the atrium and the ventricle with four-chambered heart and septation of pulmonary artery and aorta. And here what happened during embryonic life in birds and humans with complete septation to four-chambered atrium. Okay. The evolution in a species repeat during embryonic development. In early stage of embryonic development, the human heart resembles fish that in each chamber is undivided and blood exists through a single aorta. In final stage of development, the human heart had four separate chambers composed of compact myocardium and blood supply by three large epicardial vessels, the right coronary and the circumflex and left anterior descending coronary artery. Here is a fish heart, and here is the heart perimordium. The prototype of the heart, they are a resemble. Sinus venosus, primordial atrium, primordial ventricle, bulbous cordis, or conus arteriosus in fish. Here is a bulbous cordis, and here is a single outlet valve. The single outlet valve will remain in the fish to the left as a single outlet valve, but in humans, in late in development in humans, it will separate it into aortic and pulmonary with aortic vestibule behind the aorta and infundibulum behind the pulmonary artery. And complete septation will occur now. Here is reptile heart with partially separated ventricle, means as a ventricle, as two ventricle with ventricular septal defect, and human embryo heart has the same partially separated ventricle. And circulation, even the circulation in the fish resembles the circulation in the human embryo. It's bilaterally symmetric. It is the same in early development. Okay? But if you don't believe me, have a look at this. It also happened in the embryo of a dolphin. In the embryo of a dolphin, limbs appear and then in the early stage and then go away. Because the dolphin ancestors used to have hind legs, were made them, but it were useless through millions of years of evolution. It means that dolphin come out into land only to go back to the ocean. So dolphin used to be a land mammal. So the history of our evolution all happened here in the womb of the mother, womb of the mother. Here, from right to left. Over several million years, mammals have evolved from other classes of vertebrates, including reptiles. As a result of this long evolutionary process, the human heart may suspect what we call it ataphism. So what is ataphism? It's the reappearance of characteristics of some remote ancestors that have been absent in these generations. Like this, the appearance of accessory nipple, appearance of a tail, or Darwin tubercle. Here is another photo of accessory nipple. And here is a web fingers. A web fingers. Oh, a severe form of web finger. Even it can happen to celebrities, like that famous actor. He has this whip in his toes. This is because the lysosome that causes the lysosome that release digestive enzymes that separate the toes and fingers does not function. Okay. And the famous penguin who has these unseparated fingers, web fingers. Also, it can happen in congenital heart disease as the myocardial architecture resembles those of reptile's heart. We call it non-compaction cardiomyopathy or spongiform cardiomyopathy, resemble reptile's heart. Or coronary circulation resembles those of reptile hearts. This is ataphism. Okay, do you remember the the embryonic disc, the three layers that surrounds by water. It surrounds from the top by amniotic fluid and bottom by yolk sac. Let make let's us make the folding. As we make the folding here, the yolk sac will be completely enclosed in the gut cavity. And the parietal layer, when it goes to make the folding, it will bring with it the amniotic sac. And eventually the amniotic sac will surround the whole embryo at the end of the pregnancy, completely surrounding the embryo, except in one place, it is the attachment of the umbilical cord to the mother's placenta. So the yolk sac become constricted in all sides and become a small sac connecting to the midgut by a narrow vitelline duct and become smaller. Here is the three layers, ectoderm, endoderm, and mesoderm, cranial end and caudal end. Here is the lateral and transverse plane, ectoderm, endoderm, mesoderm. And here is the notochord that will form the neural canal. And here is the subdivision of mesoderm. We have the axial mesoderm, notochord in yellow, and we have para-axial mesoderm, intermediate mesoderm, and lateral plate mesoderm. Lateral plate mesoderm will split into two layers, the parietal one and the visceral one, like here, and it will enclose the yolk sac like this. And the parietal layer, as I mentioned before, will bring the amnion with it and completely surrounding the embryo except at the umbilical cord. And here will be the embryo with the heart here and the amniotic fluid and the yolk sac. Here is the midgut connecting to the yolk sac. Connection of midgut with yolk sac is reduced to the yolk sac or vitelline duct. And here is the embryonic gut tube, and here is the yolk sac, and here is the stalk. And here is the intra-embryonic coelom. After folding occurs, the embryo lies within the amniotic cavity. Ventral body wall closes completely except for the umbilical region where connecting stalk and yolk duct remain attached, like this. If we have a cut section, we can see on the right that amniotic sac completely surrounding the embryo, but at the region of where the umbilical cord will be, it's not surrounded it. So understanding the development of a heart can be very confusing because hearts are three-dimensional structures that change their shape over time. So let's do the cardiogenesis in illustrations. This is in right in red. This is the cells for cardiogenesis will form the heart. I painted with red color. It will be in the splanching lateral plate mesoderm or visceral mesoderm. Let's have a look what will happen. Here at the two sides, there are the cardiogenic cells that will form the heart. So they're gonna move with the visceral mesoderm. And actually, the endocardial tube began to develop in each side of the splanching mesoderm, and they will fuse together. Bilateral folding brings the two endocardial tube together. And the two endocardial tube fuse to form the heart primordial. And the parietal layer will follow. So here will be the gut, and in front of it will be the heart, like in adult arrangement. This is the heart primordium, a single tube. So the tube remained attached to the dorsal part of pericardial cavity by mesodermal tissue called dorsal mesoderm. Here is the dorsal mesoderm. This mesoderm disappeared to form two pericardial sinus, the transverse and the oblique pericardial sinus. Craniocaudal folding. Here is the cranial end of the baby and the caudal end of the baby or the embryo. The cardiogenic area is in front of oropharyngeal membrane and the neural plate. Here is somatic mesoderm and here is splanchnic mesoderm. and the heart tube will appear here. Future heart will develop in the splanchnic mesoderm. An area of blood-forming cavity is established in visceral mesoderm in anterior part of the embryo. Here is the future heart. Here is the future pericardial cavity. Here is the future mouse and the future brain. As we see here, the heart is anterior and the brain is posterior and the mouse is posterior. And this is not the arrangement in adults. So it has to fold. Look here. Here is the primordial heart in left. Is primordial cavity for the pericardium. And here's septal transversum. So the septal transversal will form the future diaphragm. And intraembryonic coelom will form the future pericardium, and here is the future. Beautiful illustration. Okay, what is the meaning of primordium? It is the beginning or the first identification of organ or structure. As here, the primordium of the upper limb that appear as the day 26. So here is the primordium of the heart, locating in the cranial end of the embryo. So to go to the thorax, it has to fold. So lateral folding will bring the heart to the thoracic region and also will bring the mouse upward and the brain upward. Due to the growth of the brain and the cephalic folding, the heart and the pericardial part of intraembryonic coelom move to the thoracic region. And here is the pericardial cavity become in front of the heart tube. And the future mouse will be in front of the heart, upward. And the brain very superiorly or cranially. And the cloacal membrane will be dorsal. Here's the oropharyngeal membrane, and here is the cloacal membrane, and the heart bilateral folding get to the thoracic region. This will rupture to form the mouth, and the cloacal membrane will rupture to form the urogenital and anal openings. So craniocaudal folding brings the heart into thoracic region, here from left to right, while lateral folding brings the heart tubes, the two heart tubes together, and eventually fuse to form a single heart tube. I'm coming to the end of my lecture. I hope you like it, as I always do. What comes next, whatever it takes to do it. Amr Zahir, the gentleman of medicine. Hmm?